Okay, this chapter is the sense organs. The first thing we need to talk about are receptors. A receptor is a structure that is capable of work as a transducer, meaning it will receive different types of energy, such as sound and vibration, for example, and will transform that into a nerve stimulus that eventually is going to be transferred to the brain. What type of information can a receptor transfer to your brain? It can transfer, for example, modality, such as, whether the information is hearing or taste or vision, location, the area where it's coming from. It's coming from your hand or it's coming from your back, as you can see in that figure. Intensity, if it's loud or soft, and duration, whether it lasts, 10 seconds or 10 hours. Now, obviously, the receptors can have multiple different types of classifications, such as based on the modality. They can be receptors for temperature, receptors for pain, receptors for light, receptors for chemicals, etc., etc. Now, we also have based on the origin of the stimulus, which can be stereoreceptors, interoreceptors, or proprioceptors. The muscle spindles are proprioceptors at the level of the muscles, as you can see right here. Another type of classification is general or special senses. The special senses are vision, hearing, etc., etc. And general is the one that we have all over our body, especially in our skin, like for pressure, temperature, pain, etc. Next topic is the general senses. We said that the receptors for the general senses are those located all over our skin. And they are going to transfer different types of information, such as heat and temperature and pressure. There are two main kinds or two main groups. As you can see here, these ones have capsule, right? This has capsule. And these ones don't have capsules. You see this one and this one right here. So these receptors are going to be grouped. One of them is going to be unencapsulated, as you can see right here. And those are going to be called free nerve endings, and encapsulated, as you can see right there. You can skip all that if you focus on this table, because in this table you have a nice summary of what they do and what they are. Right here you can see unencapsulated and encapsulated. For example, this one says free nerve endings. What do we use that for? For example, pain. This one, tactile corpuscle. What is that one for? Light touch. You don't need to know the locations specifically, right? As long as you know that we have two main types of receptors for general, that would be more than fine. So if we keep scrolling down, somatosensory pathways, and they are using pain in here as an example. Right here in the finger, you're going to stimulate the receptor. The receptor sends the information up to the brain using the sensory nerve, which you have right here in red. Now, the only part that you need to pay attention now is that, as I explained before, it's not just one nerve, but there's actually three. Here you have the first nerve gets in contact with the second nerve in the spinal cord right here. It makes a synapse. The second nerve goes up, makes a synapse in the thalamus, and the third order neuron is this one that takes information up to your brain. We scroll down. There's something called referred pain, as you can see here. This happens because when people have heart attacks, they feel pain in the elbow because the first order neuron for the skin at this level goes this way and makes a synapse with a second order neuron. But what happens is that the heart, the first order neuron, also makes synapse with the same second order neuron. So when the brain receives the information, it's not sure whether it's receiving the information from the skin in the left arm or in the heart. So that's why you feel pain in multiple different areas. That's called referred pain. CMS modulation, how do you modulate pain? Endorphins. In this case, it's heat. So you can see here you have the receptor. The nerve is going to be in red, sensory nerve, and sends information to your brain. So we scroll down, and that's about it for this part. Next topic is chemical senses. This is what we're going to see as special senses, such as, for example, taste. Taste, the sense for taste is obviously, as you know, located in the tongue. The tongue is not smooth, right, like the screen in your computer, but rather has a whole bunch of rough surfaces, because in your tongue what you have are going to be papillae. You see this? These are going to be papillae. Inside the papillae is where you have the taste buds. Okay, these are the taste buds right here. You have a nice picture in here of the papillae. See, this is the papillae, and inside you have the taste buds. Do we have different types of papillae? We have valate papillae, fungiform papillae, and foliate papillae, as you can see right here. Foliate, fungiform, valate, and filiform papillae. The papillae, you may assume that all of them have taste buds, but they do not work. Taste buds that we have are located, at least most of them are located in the valate papillae right here. There are two main types of taste buds. One of them is going to be for flavor, and the other one is going to be for texture. That's why you can distinguish, for example, whether you are eating ice cream or if you are eating popcorn. What type of flavors we can identify? As you can see here, at least in your book, salty, sweet, sour, bitter, and umami. Those are the main flavors. If we scroll down, projection pathways. This part is showing you all the cranial nerves that work with the tongue. We see here we have cranial nerve number 7, number 9, number 10. So those are the ones necessary to send information up to your brain from your tongue so we can receive information regarding taste. Smell, in this case, the receptors are located inside your nose, and the nerve that is going to help take the information up to your brain is going to be the cranial nerve number 1. So basically what happens in here, other particles that are in the air, they go inside your nose and they are being depicted here in green. So these other particles are going to get trapped in the mucus that we have in our nose. When they get trapped in the mucus, these other particles dissolve and they release the chemicals that they have. And as you can see here, these cells, they are going to be the receptors that are going to capture these chemicals that are going to be dissolved in the mucus, and they transfer the information up into your olfactory bulb first. From the olfactory bulb, the information is going to be transferred to the area in your brain where we are going to process the information regarding olfaction. So if we can see that right here, you can see the brain. From your nose, the information goes up to your brain and it reaches the olfactory bulb right here, yellow. And then from there, the information is going to go down to different areas in your brain, because remember, as I mentioned before, smells are usually associated with images or sensations. And when I told you that if you smell gravy and turkey, that brings memories about family and your best friends, etc., because they need to be associated with other areas. That's why you see right here the primary olfactory cortex right here in brown, but you see they are associated with other areas where sensations will be stored together with these different types of smell. So that's about it. Okay, the next topic is hearing and equilibrium, and we scroll down right here, and obviously we need to talk about sound. Sound is just the vibration of molecules that we can hear. That's what sound is, and these vibration molecules can be achieved by by a speaker or by our vocal cords, etc., etc. Now, sound has certain qualities such as loudness, but I'm not going to talk about any of that. That you don't need to know at least that part for our class. And if we look at the anatomy of the ear, we're going to see that we have an outer ear. That's what we see in this figure. And we have the lobule right here at the bottom. We have the helix. And we have this meatus, which is basically the cavity where the sound is going to go inside our ears. And this is called the external acoustic meatus. We can see in this other figure that the ears are divided in 3 pieces: outer ear, middle ear, and inner ear. The outer ear starts right here. Right, these areas that we said, this is external acoustic meatus or auditory canal. It goes inside, right, and it ends in the tympanic membrane. The middle ear goes from the tympanic membrane to where this structure right here, which is going to be the vestibule, is going to start. So if we talk about the middle ear starts basically in the tympanic membrane, continues with these 3 little bones. They are called malleus, incus, and stapes. All 3 little bones are called ossicles. And in the middle ear, we have this very important structure, which is going to be the auditory tube that goes down into basically the back of your mouth. Helps release air pressure that we may get right here in this area. So that is the middle ear. Then we have the inner ear. In the inner ear, we have 3 main structures, basically. We're going to have in here cochlea. This is the vestibule. And this one right here are the semicircular canals. The cochlea is going to help you with hearing. Vestibule and semicircular canals are going to help us with equilibrium. As you can see here, these yellow areas are going to showing you the nerve, which is going to be cranial nerve number 8, the vestibulocochlear nerve, that is going to help us with hearing and equilibrium. If we keep scrolling down, we can see the middle ear in there that we already covered. We said we have the 3 little bones, malleus, incus, and stapes, and all 3 are called the auditory ossicles, as you can see right here. And also don't forget that we have a very important tube that is going to help us release pressure, which is called the Eustachian tube, also called the auditory tube. In the inner ear, we said that we have the one that is going to help us hear, cochlea, and other two, they are going to help us with equilibrium, the vestibule, and the semicircular canals. We scroll down, the cochlea. You can see a cut of the cochlea where you have 3 tubes, and each tube is filled with a fluid. 2 right here in green, they are going to be filled with perilymph, as you can see here. This one on the top is going to be called scala vestibuli. The one on the bottom is going to be called scala tympani, and both of them, they are green because both of them are filled with perilymph. The one in the middle is called the scala media, or the cochlear duct, and this one is filled with endolymph, as you can see right there. In the scala media, you can see with this arrow, we have the organ of Corti, where you find the tectorial membrane, and you have the hair cells. The hair cells are going to be responsible for sending the information to your brain to hear.